Okay, good morning, everyone. Thank you for this opportunity to speak. So I think my talk's going to be a little bit different. I'm going to be talking about our TB tissue repository and why we need linked data in our lab at RE. So what is RE? We're basically an HIV and TB research institute based on the east coast of South Africa. We have two different campuses. I'm from the Durban campus, which is where we have our basic science labs. There are about 8 groups there. I'm from the Stain lab, and we have a lot of modern equipment to study various aspects of HIV and TB. We also have about 200 kilometers north of us a population study at a place called Somkele near a town called Matuba. And this is a longitudinal study following about 100,000, I think it's closer to 200,000 people now with HIV and TB, various aspects, social aspects and clinical aspects as well. And this will be important for what I'm talking about later for a couple of reasons, which I'll mention then. We were able to tap into that. So a little bit about TB itself. It was briefly supplanted by COVID, but it's currently still the deadliest infectious disease. About 11 million people are infected every year, and about 1.5 million people die. And the incidence and prevalence in South Africa is very high. About 280,000 people get infected, mortality of about 54,000. And this is complicated by co-infection with things like HIV. It can be treated, but the treatment is very arduous. It takes a few months. There are some newer options available now, but it's still very difficult to deal with. And there's also the problem of drug resistance. So this is a problem of major public health importance in South Africa. And something that's going to be relevant for the data I'll be talking about later is the clinical spectrum of TB infection. So for those who aren't familiar, TB is caused by a bacterium called Mycobacterium tuberculosis. You breathe it in, it settles in the lung and starts making a home there. It infects the macrophages, which is a part of your immune system. Now, initially with your innate immunity or your acquired immunity, you may deal with the disease and resolve it. But it can progress into what is called latent TB or subclinical TB. There is a little bit of a dispute about what to call this within the community now. But the point is that it can be cryptic. People can be infected, but otherwise asymptomatic, and you may not know that they have TB until an autopsy after they die. And from then it can progress onto active TB disease, where you start seeing a lot of destruction of the lung. And what we're doing in the Stain lab is we're trying to study TB in its totality. We're not just looking at the lung, we're looking at other organs, and we're using modern day techniques to characterize the TB lesions. And we're also looking at disease states beyond the typical granuloma that is caused by TB. And we're doing this by going back to looking at the human tissue. So this was pretty normal in early 20th century and late 19th century, was to look at TB from autopsies in human lung. But autopsy rates gradually declined over the course of the 20th century. This happened all around the world. At the same time, good antibiotics were found for treating TB. And so it got less attention, and people also switched over to animal models because they were a bit more convenient to work with. The problem with animal models is we don't always see all the phenotypes that we see in human TB. So at RE, we're fortunate in that we are able to go back and look at the human tissue. The reason for this is that we have collaborations with local academic hospitals. So because of those collaborations, we are able to get human tissue from lung resections. So these are living patients where the doctor has recommended that part of the lung or the whole lung get removed to help them get better from the TB. We also get specimens from autopsies from those hospitals, from the forensic and anatomical pathology departments. And the first reason I mentioned the Somkele cohort is we now, in our lab, we have an autopsy program. Part of our team, we have 2 members. One is a forensic pathologist and a prosector. And within that cohort, if somebody dies, they will offer to do a free autopsy if the family wants one. And if they consent, we can get those lung samples as well. But they'll do the autopsy anyway, even if the family doesn't want to release the samples. In addition to that, our PI has a lab at UAB, University of Alabama, Birmingham. And there we are able to get post-mortem samples from the US. So we are able to get samples from a low incidence country and a high incidence country. And this also gives us another opportunity to look at samples that may have subclinical or latent TB. And it's because of this that we have tissue samples from about 900 different patients. And it's from this is basically where all the data that we have is originating. In addition to that, TB is a complex disease. So these are 2 lung portions from 2 different patients. And you can see, let me get the laser going. There are various different pathologies that can happen in advanced TB. You can get calcification. I think that's a calcification there. You can get caseous necrosis. You can get fungal infections once you've coughed the necrosis out. That's otherwise called a mycetoma. You can get cavitation, wall thickening. You can also get healthy tissue. And one reason that this is important, within the same lung, you can see many temporal states of a TB infection. And what we are trying to do is understand better how the TB progresses. This is just an example of how TB can look under a microscope. So in the first example, this is a Zeal-Nielsen stain. And these little purple rods, these are the individual bacilli. This is a hematoxylin eosin stain. And here you can see a necrotic granuloma. And this is an immunohistochemical staining for a cell surface antigen. And that's what you see in the brown here. And the hematoxylin is used to stain the nuclei. And what we're trying to understand is how do these early lesions progress? We're trying to understand this at a cellular and a molecular level, because at this level, there's some kind of decision point. A lesion may become regressive and get resolved, or it may become progressive and trigger a severe inflammatory reaction to the necrosis and cause a lot of damage to the lung. And this is where the Delta Tissue program came in. So I don't know if anybody here is familiar with the LEAP Foundation. This is a very unique funding opportunity, and we were successful in becoming a part of this. And what it did was it brought together all sorts of different people who we otherwise wouldn't have been able to interact with, to study these various diseases at a molecular level using very modern techniques. And what they wanted to do is come up with a so-called tissue time machine to be able to predict forwards and backwards what's going on within the disease. 3 diseases were selected for this: glioblastoma, or GBM, triple-negative breast cancer, and tuberculosis. But it also brought together instrument developers, both from academia and industry, as well as people from the software data analysis side, image analysis and data integration experts. And this is how I met Andra, and it's somewhat accidental that I've ended up at a conference speaking about linked data. And this is a photo from one of our hands-on meetings at Cape Town last year. Now we were a bit late to the linked data story. Andra kept banging on about it, and it took us a while to understand why this is important. So we only started this last year. But later on, you can hear from Tanya Garcia from the GBM group. They got onto this much earlier, and they're much further along than we are. So to get into where our data is coming from itself, this is what our tissue processing workflow looks like in a very brief summary. So in the first step, we have this macropath table where we photograph the samples and label them. So that is Finessen Naidu, our forensic pathologist, and that is Kapongo Lumumba. He's a jack of all trades and helps us with the data curation, as well as autopsies and a lot of other things. This is Curation Naidu, and he is our histologist and histolab manager. After the samples have been labeled, they can be scanned by the micro-CT scanner. So we have a micro-CT scanner which allows us to look at the tissue in 3 dimensions. This is what I do most of the time, and I ended up landing in this role as well. Then the tissue is cut up into smaller samples and blocked and converted into these wax blocks. The blocks can be scanned again, depending on what we want to do. But then from the blocks, from a single sample, we can get hundreds of blocks, and then from In individual blocks, you can potentially get thousands of glass slides, and each of these can be imaged using various modalities. So at each step, there's data and metadata being generated, and there's also potential amplification in the amount of data that is coming out. Now, the way we're capturing our metadata is using REDCap. So the first data that comes in, we've got the data from the macrophage table. We also get some data from the hospitals, some patient info, things like age and sex. The amount of data we get is a bit variable. We're not in complete control of that. We'll sometimes get a treatment history, whether they were HIV negative, things like that. There might be other comments from the doctors themselves. And in REDCap, we have forms for capturing various different stages of the process, the patient information, organ information. Then as the organs are divided up, then from each of those, the paraffin blocks, the glass slides, micro-CT, and histological appraisal, and so on. And for those who aren't familiar with REDCap, I think it stands for Research Data Capture. It's a fairly mature system for designing forms for capturing this kind of information. You can build a lot of logic into it. One of the reasons I'm mentioning it is this is being used to capture all the data from that population study of 100 or so thousand people in Matuba. And at RE, there is a team that is very capable and knows the system very well. And it's fairly versatile. You can also use it in the field on a tablet. You can use it in offline mode. And in our lab, we've barely scratched the surface of what we can do with this. So this is just an example of what the forms look like. We have a form for the histological appraisal and for the blocking and the cutting initial slides and so on. But then we also have the imaging. So this is what some of the micro-CT imaging looks like. So we'll initially scan a sample without any kind of treatment before blocking. And sometimes we see things like this. We can see these calcifications within the sample, and this can help us scout for what we want to look at closer next. We also can stack the blocks in this 3D printed holder, and these bright spots are the calcifications. And this helps us decide, okay, which block do we actually want to cut? How far do we want to go down? And sometimes we can see intermediate opacities that correlate to other lesions within the TB. We can also use a contrast agents to stain the samples, and this allows us to see a lot more of what's going on inside when we do the micro-CT scan. And we can do some fancy segmentation. So in yellow here, you can see these necrotic lesions, and this is how we initially found that these lesions can form these very, very complex shapes. In many depictions of a granuloma, you'll see it's just round or circular, but these things can actually become very complex and ginger root-like. So this is the one source of data that we need to capture. But thanks to Delta Leap and the other imaging we're doing, we have a lot of other modalities that we've been getting image data from. So this is a normal H&E. One of the instrumentalist groups that we worked with in Delta Leap was the metallomics group from Alex Morel at UCL. So in this case, you're seeing the same lesion imaged with H&E, metallomics, and multiplexed immunofluorescence. With Jeff Jones at Caltech, we've been doing higher resolution proteomics, and with Hal Pasanga, we've been doing spatial proteomics. And from this comes lots and lots of data that we need to try and tie together. We've also been using various OMERO servers to do collaborative annotation with Sanger, so they can know which ROIs to take a look at. So this is where Thnessen and curation can come in, and they can do annotations either in OMERO or QPath, whatever turns out to be convenient. And then this is another case of data that needs to be linked together with everything that's been generated. We're also looking at automated segmentation. So here are some examples from the Halo software that Compango has been working with. So we would like to generate lots of these kinds of ROIs of lesions and various other types of tissue to start getting statistics from that. And in this case, this is some automated segmentation I've been trying out on 3D in the micro-CT. And ultimately, we would like to pull these ROIs and link everything together with the rest of the repository. And one problem that we face is that as this data grows, you get your initial systems of handling it get rather cumbersome. So Once upon a time, we had less than 100 samples. It was fine. We kind of knew where everything was. You could use the PIDs that came from the hospital or had some rather quixotic internal names and kind of know where everything is. But very quickly, we ended up with a case of too many folders, and partly because of getting to meet people like Josh Moore through Delta Leap, we set up our own OMERO server last year. And we're setting it up in such a way that it make it easier to search our image database. So at the moment, we have some tags and key value pairs for fast searching. So this is intended for other people in the lab, like my PI or the pathologists, to do fast searching. We are able to import all the annotations that we've accumulated in the old .ndpa files. For those who are familiar with the Hamamatsu platform, their software saves annotations in these files, but we can import those into OMERO so that all that work doesn't get lost. But a problem that I'm facing now is what I call the inertia of familiarity. People are used to going back to their old emails, their old folders, looking for things. Now it's a problem of trying to convince them to use it. This is an example of me testing the annotation import. But ultimately, we also want information to flow from our REDCap database into this. We can't just do all the tags manually. We need a way to link all that, things like age, where the sample came from, things like that. So how are we trying to tie this all together? So first, we have to summarize as REDCap, where we're storing patient info, organ and sample info, blocks, and appraisals, and things like that. Then we start getting into the imaging. So we have the whole slide images, which go into OMERO. And with Andra's help, we are linking these together via an RDF export. And we're able to link this together, but one of the things I'd like to do next is to get information from REDCap to flow back into OMERO as tags or key value pairs via this RDF linkage. And then we have the various images. Currently, a lot of our histopathology images are on our OMERO server, but we also need to add our metallomics and various other modalities. It's not that suitable for the micro CT, so we'll probably need a different platform for that. And then we have the manual annotations and the auto annotations, and then we get all these regions of interest in /ROIs from that. Ultimately, we'd also like those annotations to go back into the RDF so we can construct queries using those. So we can do more complex queries instead of just where did something come from. And eventually, we'd like to feed this into some kind of AI or machine learning so that eventually we can maybe stop asking questions. But I think that is unlikely. So just to summarize, we're using REDCap to capture patient metadata, samples, slides, and things like that. We have an OMERO server that's used to browse and search imaging output. So far, we only have manually-curated tags and key value pairs. But we have a proof of concept by being able to link REDCap and OMERO with REF, and ultimately on top of that, we want to have a semantic model to enable more complex queries. And this is an example of a query we can do so far. So in this case, this number 5 is just a number that's internal to REDCap, and it refers to samples that come from the lab at UAB, University of Alabama. So obviously we need a mapping to make that more human readable. And in this case, we see a list of some of the images, it's the first images coming from samples from UAB. But obviously we have a lot more that we need to do on top of this. So ultimately we want to link REDCap metadata with outputs from all the imaging platforms so we can query across them, including all the patient metadata, things like the histology, what stain was used, what pathologies are present in the images by the various modalities with the ROIs. And we want to reach a point where we can use this to feed queries into machine learning tools for automated segmentation and so on. So to conclude, we have a rather cumbersome system that we had before is being replaced with this linked OMERO REDCap. As far as we know, this is the first example of linking REDCap and OMERO with a semantic model, an attempt to do so. We're in the process of creating a boxology to put on top of this, and ultimately want to include as many outputs as possible. And something that's maybe fortunate or unfortunate, our lab was awarded an NIH R24 grant to provide access to this tissue repository, and we will need something like this to allow us to do our own internal queries and to make that access proper for outside scientists as well. So quick thank you to all the funders and to everyone who's been involved and more with this work at some point along the way. So I don't know how I'm doing for time. If there are any questions. Yeah.
No, I think this kind of cross-modality interoperability is going to be key in the future, to make the possibility to query across images or genomics or clinical data.